Okay, guys, so lab 45, okay? Here are the objectives and let's get started. So, the respiratory system is going to consist of passages that will actually filter and moisten and warm the incoming air and transport it into the body, into our lungs, and to the many microscopic air sacs where gases are exchanged. So the respiratory system is going to provide oxygen for aerobic reactions and eliminate carbon dioxide at the appropriate rate to maintain the internal environment pH. Okay. So the organs of the respiratory system can be divided into two tracts. We have the upper respiratory tract, and then we have the lower respiratory tract. So the upper respiratory tract is going to consist of the nose, the nasal cavity, your sinuses, the pharynx, and the larynx. Whereas the lower respiratory tract is going to consist of the trachea, the bronchial tree, and your lungs. Okay. So here, let's first start with the nose. So your nose, basic, also known as your external nares, is going to provide opening for air to enter and leave your nasal cavity, and it's going to be openings are protected by the internal hairs. Okay, the nasal cavity is this hollow space behind the nose, and it's going to separate into left and right portions of the nasal septum. Okay, by the nasal septum, sorry. And the nasal concha or superior, inferior, middle, these turbinate bones curl in from the lateral walls of the cavity, and the concha is a concha. It's going to separate the nasal cavity into these passageways called the meatuses. Okay. And so the upper portion is going to contain up here, the upper portion is going to contain olfactory receptors for sense and smell, and that's going to be lined with a mucous membrane, specifically pseudostratified ciliated epithelial tissue. And the nasal cavity in general is what's going to function in that conduction of air and warming and moisture and filtering any incoming air we're about to breathe. The nasal concha actually supports the mucous membrane that lines the nasal cavity, and the concha also helps increase the mucous membrane surface area. Okay. So here with the nasal cavity, this is that pseudostratified um ciliated epithelial tissue I was talking about earlier is going to contain goblet cells, okay? Um, and these pseudostratified ciliated columnar, sorry, these are columnar, pseudostratified ciliated columnar epithelial tissue is going to contain goblet cells that's going to be interspersed throughout the tissue to help produce mucus. And the particles and microorganisms from inhaled air actually gets trapped in the mucus. Okay, and what happens is that cilia sweeps the mucus towards the pharynx, and then that microorganism is swallowed, and the mucus are destroyed in your stomach by your stomach acids and contents. So here are the sinuses that you can see from the images being pointed with the arrows, the paranasal sinuses. And so these are basically air-filled spaces within the maxillary slash frontal and ethmoid and sphenoid bones of your skull. And this opens into the nasal cavities. And the mucous membrane lining is continuous with the lining of the nasal cavity. Then we have the pharynx, also known as the throat. And this is the space posterior to the nasal cavity, oral cavity, and the larynx. And this is the passageway for food, liquids, and air. And this aids in sound production. Now, there's three portions of the pharynx. There's the nasal pharynx, oral pharynx, and the laryngeal pharynx. Okay. So then here's the larynx, and the larynx is this enlargement in the airway superior to the trachea. and anterior and somewhat inferior to the laryngopharynx. And this is what's going to move air in and out of the trachea. And this is where our vocal cords are going to be housed at. So this is going to compose of a framework of muscles and cartilages bound by elastic tissue. So this is an anterior point of view, while down here is a posterior point of view. And so you have three large single cartilages. You have the thyroid cartilage, as you can see right here, which is the largest cartilage. You have the cricoid cartilage, which is below the thyroid cartilage. And then you have the epiglottic. And that cartilage is basically the flat-like epiglottis, part of the flat-like epiglottis. And you have three pairs of small cartilages, and those are the arytenoids, the corniculate, and the cuneiform cartilages right here. The corniculate, arytenoids, and the cuneiform cartilages. So the larynx contains two horizontal vocal folds composed of muscle and connective tissue. So the false vocal cords, that's going to be your upper vestibular folds. So these are the false vocal cords right here. And this is where no sound production is going to take place. No sound production is going to happen with your false vocal cords. Your true vocal cords right here, these are your lower folds, and these are what produces your vocal sounds. The opening between them is known as the glottis. Okay, that is the glottis. Okay, the opening between them. So the trachea, also known as our windpipe, is this flexible cylindrical tube around 2.5 centimeters long and like 12.5 in diameter centimeters. Sorry, I said that wrong. 2.5 centimeters in diameter and 12.5 centimeters in length. There we go. It's like diameter is this way. So this is going to extend downward anterior to the esophagus. And as it enters the thoracic cavity, it spills into left and right primary bronchi. And it's lined with ciliated mucous membrane with goblet cells. And the wall has these C-shaped rings known as cartilaginous rings of hyaline cartilage to prevent the collapse of the trachea. Okay. So here's a cross section of the trachea to show the C rings of hyaline cartilage to keep it open, and the pseudostratified ciliated columnar epithelial tissue that lines the lumen. So a tracheostomy is a procedure that cuts an opening in the trachea to insert a tube for air exchange. This is done, for example, if an object is lodged into the larynx or the trachea. And so the bronchial tree, that's going to consist of this branched airways leading from the trachea to like the microscopic air sacs within our lungs. And so the main bronchi, also known as the primary bronchi, arises from the trachea, and each one enters into a lung. So starting with the trachea is going to, the branches, like airways actually resembles like an upside down tree. And so with like successive divisions of the branches from the trachea to the microscopic air sacs. So that's how it would go. So so right here would be right and left primary bronchi. Then from there you would get your secondary, or also known as lobar bronchi. And then from there you would get your tertiary bronchi, also known as segmental bronchi. And then from there it would break down into the after tertiary or segmental, it would go into the intralobules bronchioles. Excuse me. And then from the intralobular bronchioles, it would go down into the terminal bronchioles. And then from the terminal bronchioles, you'll get the respiratory bronchioles. And from the respiratory bronchioles, you'll get the alveolar ducts. And the alveolar ducts, you'll get the alveolar sacs. And then from the alveolar sacs, you actually get each individual alveoli. Okay. So here you would see the alveolar duct, the alveolar sac, and then each individual alveoli. So all of the structures of the bronchial tree down to the terminal bronchioles are part of the conduction pathway since they transport air and do not take part in gas exchange. And so the structure between the respiratory bronchioles and the alveoli are part of the respiratory pathway since they actually contains the alveoli and perform the gas exchange. Okay. So the structure of the main bronchi is similar to that of the trachea, but they contain cartilaginous plates instead of C-shaped rings. And so as like the bronchi continues to branch and become narrower and narrower, and the amount of cartilage will actually decrease. And so the cartilage will actually disappear in the bronchioles. And here you'll start to see smooth muscle becomes more prominent, then begins to diminish from the bronchioles to the alveolar ducts when it disappears. Okay. So the respiratory tubes become really thin, like super thin, and the types of epithelial cells actually will change. So the pseudostratified ciliated columnar epithelial tissue is going to be in your larger tubes, but it's going to then go to simple cuboidal epithelial tissue within the respiratory bronchiole, and then it's going to turn into the simple squamous epithelial tissue within your alveoli. Okay. So simple cuboidal epithelial tissue in your respiratory bronchioles, and then simple squamous epithelial tissue in your alveoli. So here you can see the network. Okay. So branches of the bronchial tree are air passages. And so the alveoli actually provides surface area for gas exchange, as we can see here. And during that gas exchange, oxygen is going to diffuse through the alveolar walls and enter the blood, and carbon dioxide is going to diffuse from the blood to the alveoli. And so the lungs, you got these soft, spongy, cone-shaped organs within the thoracic cavity. And the lungs are separated from each other by the heart and the mediastinum. And so the right lung, okay, is going to have 3 lobes, whereas the left lung is just going to have 2 lobes. Okay. So this is a transverse section. So here, the hilum, that is going to be the region on the medial surface of each lung through which the bronchus and the large blood vessels will actually enter. But here, you can't really see the hilum here. It's just basically the medial surface. So the hilum would be like right here, but you can't really see it as well. It's just the side that where the blood vessels in the trachea enters the lungs. The visceral pleura, which is going to be in green, is the inner layer of serous membrane that's attached to the surface of the lung, whereas the parietal pleura is the outer layer of serous membrane, which actually will line the thoracic cavity. And then the pleural cavity is the potential space between the visceral and parietal pleura. Okay. So then this is just a chart explaining parts of the respiratory system. You can read that on your own. Now you're going to have a spirometry experiment. Okay, a spirometry experiment where it's a virtual lab simulation. And so I want to talk a little bit about respiratory volumes and capacities. Okay. Um, so you can get an idea while you're doing the virtual lab simulation. So different degrees of effort in breathing moves different volumes of air in and out of the lungs. And so this measurement of air volumes is called spirometry, okay? And so in that virtual lab simulation, you actually perform spirometry on several patients to perform pulmonary function tests and diagnose pulmonary diseases. Now, some air entering respiratory tract during breathing does not reach functional alveoli. These are called dead spaces, okay? And so anatomical dead space is basic, or yeah, anatomical dead space is the air in the respiratory tract that remains in conduction um, structures and does not reach the actual alveoli. Whereas the alveolar dead space, that is this a part of the respiratory tract that reaches the non-functional alveoli, such as like alveoli associated with capillaries with poor blood flow. This occurs occasionally. And then there's physiological dead space, which is the total of anatomical and alveolar dead space. And so under normal circumstances, anatomic dead space equals physiological dead space. And so that's usually around 150 milliliters per breath. And then what else I want to talk about is the minute ventilation and the alveolar ventilation rate. So the minute ventilation rate is the volume of new atmospheric air moved in the respiratory passage each minute. So it's abbreviated MRV. And so MRV is going to equal this tidal volume times the breathing rate. Okay. Over the respiratory rate. Okay. Um, so MRV basically is tidal volume times respiratory rate. And since some of the new air in each breath is going to remain within your physiological dead space, another figure can be calculated to represent the actual amount of inhaled air that reaches the alveoli each minute. So it's called AVR, or alveolar ventilation rate. And so AVR is basically the respiratory rate times the difference of the tidal volume minus the physiological dead space. Okay. So this is basically the volume of air that reaches the alveoli each minute. And this is an important value since it impacts the concentration of oxygen and carbon dioxide within the alveoli. Okay. So then, so let's go over these volumes and capacities. So the tidal volume is going to be around 500 milliliters. And that is basically the volume of air moved in or out of the lungs during a respiratory cycle. So, like one cycle. And then you have the IRV, also known as the inspiratory reserve volume. That is basically going to be around 3,000 milliliters. And that's just the maximum volume of air that can be inhaled at the beginning of a resting inspiration, versus the expiratory reserve volume, or the ERV, is just the maximum amount of air that can be exhaled at a resting expiration. You have residual volume down here, and you're like, what's residual volume? So residual volume is the volume of air that remains in the lungs after a maximum expiratory effort. So it's like whenever you try to push out as much as you can, it's just the part that's still there. And the vital capacity is just the sum of the ERV, the IRV, and the tidal volume. Okay. It's just basically the maximum amount of air that you can take in after a deep breath. And then the IC, also known as the inspiratory capacity, that is basically the maximum of air you can take in when you inhale following an exhalation. And then the functional residual capacity is basically the volume of air that remains in your lungs following an exhalation from the resting tidal volume. Okay. And then the TLC, or the total lung capacity, is just the total volume of air in the lungs that can hold. Like, it's just the total amount of air you can hold in your lungs. And that completes going over the respiratory volumes and the capacities. Okay, guys. Stay safe and study hard.